All right, we are all set. Uh, okay, hi everyone. Welcome and thank you for joining us for Vector Builder's Gene Delivery Seminar Series. Our speaker today is Kathy Krentz, the director of the Animal Models Core at the University of Wisconsin. After completing her bachelor's degree from the University of Wisconsin River Falls, Kathy received her master's degree at Virginia Tech before beginning a more than 30-year career at the University of Wisconsin's Transgenic Animal Facility, now Animal Models Core. There, Kathy manages everything from database management and grant writing to microinjection, cryopreservation, and other knock-in techniques that we'll be learning about today. So following the presentation today, we're going to be having a Q&A session. So please type any questions that you have into the Q&A text box at any time during the presentation, and I'll read them out at the end. And now I will hand things over to Kathy.
Thank you, Connie. And a special thank you to Vector Builder for allowing me to be here today, present the work that we do here in Wisconsin, and to give a little insight into how we are doing gene delivery into rodent animal models. So as Connie said, I work as the director of the Animal Models Core. The title of my talk is Advancing Gene Delivery of Large Knock-in for Rodent Animal Model Production. So my core is located in the Biotechnology Center. We are a fee-for-service facility, which means we charge labor and supplies back to labs that we work with. Our whole department is all core facilities that do the same in terms of charging back for labor and supplies. As Connie said, I've been at the UW for over 30 years. I've been the director for around 10 years. I have 3 full-time staff that help with all of the embryo manipulation, all of the breeding of the mice and the rats, and all of the work that we do for labs on campus. And we employ 1 undergrad student who helps us. My core is also part of the NCI Carbone Cancer Center Shared Resource Grant. They give some salary support for all of my staff. And our department is part of the Office of the Vice Chancellor for Research. I'm very lucky that our department has multiple cores that we work closely with. There's a gene editing core, and they work in tandem with me to design all of the CRISPR reagents, and they do all of the genotyping. There's a DNA sequencing core in my department, and so all of our animals are sequenced in-house. There's a bioinformatics team that helps with all of the sequence analysis. And so, as I said, I'm very fortunate here at Wisconsin that we have everything within one department. So most of my talk today will feature talking about CRISPR-Cas9, how we use that to design and create animal models that are used for mainly biomedical research. We do a lot of close work with the UW hospital and doctors that have patients with specific genetic mutations, and then we mimic that within an animal model. I just wanted to make sure everybody understood. I don't want to go into a lot of details about CRISPR, but we can design guide RNAs that target a specific region of the genome. We can use Cas9 nuclease to go in and cut a specific cut site close to the PAM sequences. This type of CRISPR projects are about 50% of the revenue that my core has within a given year. My core also does such things as cryopreservation of important animal models. We can freeze them down and at any point in time bring them back. We also do importing and exporting of frozen samples all over the world, and we do rederivations to clean up pathogen contaminated animals. So for CRISPR animals or knockouts or knock-ins, we can take the gene of interest that a specific lab has. We can easily create an indel or a genomic break in the DNA. We can delete a specific gene, or we can add a reporter tag, such as GFP. We can make a tissue-specific Cree model, or we can even add 2 LOXP sites and create a floxed mouse line. And so this is all routinely done in my core. We've been doing CRISPR probably now close to 10 years with varying degrees of techniques over the years. And as I'm sure many people are aware, CRISPR has really evolved over the past couple of years. And there's always new, better ways and faster ways to do this. And we routinely are using CRISPR to make sure everybody's on the same page. When I talk about pre-implantation embryo development, 1-cell embryos are roughly 12 hours post-fertilization. And that's the majority of what we work with in my core, is collecting and microscopically sorting good quality 1-cell embryos. Within a 24-hour period, those embryos will divide into 2 equal uniform blastomeres, and we call those 2-cell embryos. And those can continue to divide and eventually develop into a blastocyst. A blastocyst is close to being ready to implant. You can see some of these embryos within, if I can use a pointer, I'm hoping you can see that. This embryo here is hatching. It is leaving the zona pellucida, which is the outer membrane of an embryo. It's leaving that zona pellucida, and it's ready to divide, ready to implant within the uterine environment. And so it's critical when you want to create a genetically engineered or genetically modified animal to get the new genetic material into the embryo early, so that every cell in the body incorporates that new genetic material. And again, so the majority of my work, we use 1-cell embryos to introduce the CRISPR material. We do a lot of mouse and rat work. I would say probably maybe 80% of what we do is mouse work, and about 20% of what we do is rat work. We use the same techniques basically across the strains. For mice, we typically use C57 Black 6 from Jackson Labs, and that's just based on popularity. A lot of the mice that we make are crossed to other lines, for example, a flox line that's mated to a Cree line. In those cases, we try to keep genetics the same. We have successfully used many different strains, including those listed here. We do not use hybrid strains. I know a lot of cores tend to use hybrid strains. We do not use hybrids. I think genetics is very important when you're looking at research animals, and especially biomedical research. It's important to keep the genetics the same across the board, and therefore we avoid doing any repetitive breeding if we don't have to. And so for those reasons, we tend to use C57 Black 6. I am also open to using the customer strain. So let's say a lab on campus has been doing some work with a line of mice that already has a genetic mutation. We can take those mice into my auxiliary mouse room. We can collect 1-cell embryos from them, and we can introduce a second edit into an already existing mouse line. And so what that does is save a lot of breeding, like 3 years, 4 years of breeding, so that we don't initially create the founder line on an inbred strain, and then they have to back cross it into their line of interest. We do the exact same things with rats. Typically, labs use Sprague-Dawley rats, which are an outbred strain. And we love Sprague-Dawley rats. Sprague-Dawleys, because they are outbred, produce a lot of embryos. and they're high quality, high fertilization rates when we use Sprague dollies. We have also successfully used multiple inbred strains of rats, and we're not opposed to doing that. We just got a request to do some work with brown Norway rats, which is going to be very challenging, as brown Norway fertility is an issue, embryo production is an issue. So, but we are up for the challenge, and we're going to try to do some work with brown Norway rats in the near future. The same holds true with using a customer strain. We had a few labs that have a rat line, and they wanted to introduce a second mutation into that rat line. And we're completely open to ideas like that. I run my core as I am here for not only the labs on campus, we also do work for off-campus labs. And I'm here to produce animals that they can use. And let's be open and have conversations as to what the long-term goal is and how can we best achieve that for the labs. My animal facility is located here in the Biotechnology Center. We are ALAC accredited. We are also an SPF1 facility, which means that we have no pathogens, no viruses, including norovirus and helicobacter. We are free of those. And so for those reasons, I sometimes get inquiries from off-campus labs that want us to do some work for them. And so we do offer off-campus services, both for labs and for companies. And we try to assist where we can, especially smaller universities that don't have a core facility like mine. And we can help them create animal models, and have done that frequently in the past. So in the past, we have used the technique of microinjection of one cell embryos to introduce the Cas9, the guide RNA, and the donor template into a developing embryo. And so the technique is always starts the same way. The technician collects one cell embryos. One cell embryos are sorted for two pronuclei. So the two pronuclei come from the egg itself and from the sperm. One carries the maternal DNA, one carries the paternal DNA. And the idea is that you have to introduce the new genetic material into the nucleus before those two pronuclei fuse together. And so in my core, we have this inverted microscope that's shown in this picture down here. This inverted microscope is hooked to two joysticks. The joysticks are hooked through tubing into two different needles that are on the left and the right. So this picture on the left shows the holding pipette, which is used to hold the embryo in place. They are floating in media in a petri dish right now. On the right is the injection needle, and that is loaded with the mix of guide RNA, Cas9, and donor template. And so the technician then aligns the needle with the pronucleus of an embryo, physically goes into the pronucleus, deposits a small amount of liquid without overloading the nucleus, and then pulls the needle out. And so this is a very labor-intensive procedure, but we have done this for decades. This is stuff I learned to do in grad school just a few years ago. And it's still used today. And so we actively have to pick up every embryo, inject it, move that embryo to a different side of the slide, go back and pick up a new embryo. And again, this has been very successfully used to make gene-edited animals. The problem here is, as I said, we tend to use C57 black 6, which number one, the cytoplasm is then pigmented, a little harder to visualize the pronucleus. The act of going in and taking that needle through the cytoplasm, puncturing the nuclear membrane, does cause some lysing. And so we do get some cell death after this procedure. After we implant these embryos into the moms give birth. We get about 60% pregnancy rates with this technique. However, we get very poor efficiency rates when you're talking about knock-in rates. So in my facility, we genotype and sequence every mouse or rat that is born. Each of them gets an ear tag. At weaning, we take a small tissue sample, make genomic DNA. And then as I said, we have a sequencing core right in the building. We take those samples down and have them sequenced in-house. What we saw back over the years is partial knock-ins or partial inserts. And so the knock-in technology really needed some work. We were getting knock-ins, but it took a lot of persistence and a lot of embryos in order to get these founders generated. Back in 2018, this Nature Protocols paper came out and introduced us to the logo or the technique called CRISPR-Easy. And this has really been a game changer in the world of making genetically modified mice and rats in particular. So on the right here, what this paper introduced is the RNP complex or an RNP assembly, in which you take the Cas9 and the guide RNAs and make them into one complex. And you do that by heating the solution to 37 degrees for 10 minutes. And then that mix is introduced by electroporation. And so many labs have used electroporation of cells for decades. Everybody knows that you can take a cell, throw it in a cuvette with some plasmid, and zap it and get that introduced into a cell line. But this is the introduction of the new genetic material into an embryo. And it's been, like I said, a game changer in our world. And so the idea is the superovulation, the embryo collection is all the same that we've done. You thin the zonas, is the paper's methodology. And then you introduce the genetic material through electroporation. They then did some ex vivo analysis by genotyping and sequencing some embryos, as well as transferring these into recipient moms and looking at the offspring. And so their proof of principle was to first target the tyrosinase gene. Tyrosinase is, allows for the coat color of the mouse. And so a typical C57 black 6 gene, the tyrosinase is not mutated, but if they went in and targeted that gene through electroporation, and they got albino mice born. And over a series of pulses, with 6 pulses, you can see that 100% of the mice carried the genetic edit. But then, as you can imagine, as you increase the pulses to an embryo, you decrease viability. And so here, 2 pulses allowed for the highest birth rate per embryos transferred. And so then they put this into practice and went and then deleted a gene, both through CRISPR-Easy and the electroporation process, and through the microinjection process, and they show that data here. And electroporation of embryos, you get almost 25% editing versus the old-fashioned microinjection technique that I displayed for you, where you typically get about 2 or 3% editing. Okay, so as I said, this has been a game changer in the field that I'm in, and that we can electroporate without having to do the laborious microinjection technique. And so here is our setup in my core. Same thing, we collect the embryos, we sort for 2 pronuclei. Okay, I'm going to play, hopefully this works. So on the right here is a microscope. We would put the petri dish of embryos here. Here's our electroporator. It's set to a certain voltage for a certain pulse length. The electrodes are hooked to a very specifically designed slide. This slide is magnified over here on the left. So the slide has two pieces of metal made out of platinum with a 1 millimeter gap. And so the electrodes are hooked to that, as you can see here. The technician would go in and load only 5 microliters of the RMP complex. So again, this is how we create this is how we create knockouts and SNPs that are shorter than 400 base pairs. Okay, so 5 microliters of the Cas9 RMP and the SSODN, if we are doing a SNP, are loaded into the center of the slide. And then if I can get, I don't know if I need to move this. I don't know. There. So if I can show this video now. So this is a high magnification of the technician depositing the embryos within the mix, very carefully depositing them in the middle so they do not touch the metal. And then they're going to apply a pulse. Watch for the air bubbles or the bubbles to form on the side. That was one pulse, and now there's going to be a second pulse. And there's the second pulse. And now they come in and they remove the embryos from the slide. So this, as you can see, is CRISPR easy, as they have designated this. We can put up to 50 fertilized embryos on one slide and zap them all at one time. And then they take them off, rinse off the slide, reload with a new 5 microliters of the RMP mix. The rest of this procedure is all the same. We surgically implant these embryos into recipient moms. The moms give birth. The pups are genotyped and sequenced. You can see the benefit of doing this and how it's so much easier and so much faster. And so in my core, this CRISPR easy technique is highly efficient. Sometimes we get as high as 100% of the animals born carrying the edits. We often get biallelic knockouts or SNPs, which makes breeding even easier. This is reproducible. We've been doing this now for probably 4 or 5 years. It's reproducible project after project. And the mice go germline, mice and rats go germline. And so this is our go-to method for, again, creating knockouts and SNPs within my core facility. Creating knock-ins is a whole different ballgame. And so we used this paper as a reference from back in 2019. And this was our go-to for a few years to create knock-in animals. So this is stuff that's larger than 400 base pair. So we used a Cas9 that had a strict abdomen connected to it. Our donors were all biotinylated, and then this created a conjugate within the cell that allowed for an easier delivery of the DNA template. So this had to be delivered by microinjection. Again, a labor-intensive, less efficient. We were looking at less than 5% of the animals that were born that would carry in the knock-in. But even though it was low efficiency, we used this for years, created many, many animal models this way. And again, it was something we just had to be very, very in tune to. We had to deliver a lot of embryos in order to achieve the goal of generating one founder. So along the same lines, the CRISPR Ready paper came out. So this is a Cell Reports paper that came out in 2019. And not only were they doing electroporation of the RMP, which I've already showed you, has been proven to be highly efficient, they introduced the idea of exposing embryos to AAV, adeno-associated virus. And when this came out, we really shied away in the beginning. As CRISPR has evolved over the last decade, there's always new things coming out. And as a core facility that needs to deliver a product, we really wait until things have really been proven before we jump on the bandwagon and switch our mode of delivery. And so this paper was really interesting and caught a lot of people's eyes. They used 3 different projects, if you will, a roughly 700 base pair fluorescent M strawberry. Another project they did was to introduce a Cree ERT that was about 2 kb. And then the third project they did was a 3.3 kb Rosa 26 insert. And the methodology is that you collect the 1-cell embryos, you put them in the virus. After a virus exposure, you then electroporate your Cas9 and your guide RNA, like we've been doing for the last few years. And the rest of it is the same, oviductal transfer and looking at the sequence of the edits. So from this paper, the first thing they looked at is a bunch of different AAV serotypes, and 2 stood out, AAV1 and AAV6. And then the rest of the experiments were done with either AAV1 or AAV6. So the first thing they looked at was different incubation times. And you can see here, they get HDR rates that increase to a point, and then there's a significant drop off. So 6 hours of incubation seemed to be a good time frame before introducing the guide RNA, the RNP complex. And then they looked at different doses, and it looks like this got cut off a little bit, but they looked at from 1 to 10 to the 8th to 1 to 10 to the 9th. And again, they saw viability drop off at 1 to 10 to the 9th. So the idea was exposing the embryos to different time frames, doing some embryo culture, and then assessing the editing rate after a few days in the incubator. And this is one of the projects they did. They had 19 mice born. From those 19 mice, 9 carried the Knockins. So 9 out of 19, gene editing rate of about 47%, which is significantly higher than this traditional microinjection method that MyCore and others have been using. So like I said, we don't always jump on the bandwagon right away, but as I kept going to conferences and hearing about other cores who were doing this, we decided it was time for us to do this. And so this is a schematic that I stole off of Vector Builder's website. I know very little, if not nothing, about virus production, but what you can contract a company to do is to not only make the plasmid, but then from the plasmid of choice, they then create the virus particles. And the thing that we liked is their quality control and their testing that has done downstream of that production. And so this is where my relationship with Vector Builder started and has grown into a service that we do on a regular basis now. So when we started doing this was last year, at the beginning of 2023, we used AAV6. We outsourced the virus particle production to 4 different commercial sources. The first source that sent us reagents, we got no pregnancies, we got no pups. And we really spun our wheels in the beginning to get this up and working. But like I said, we just kept going to different sources and eventually landed on Vector Builder, who now makes all of our plasmids, and then from that does the AAV production. We initially tested 2 different concentrations, 1 times 10 to the 7th and 1 times 10 to the 10th. And the final decision on who to go with is based on the delivery time. So when we place an order, how long do we have to wait before we have virus particles in-house? The price, of course, is also important. And then also the success rates of creating these knock-ins. So here's my pipeline that we use here in Wisconsin. We collect 1-cell embryos, same as we do for all of the other techniques that we've used over the years. We use 1 times 10 to the 10th as our go-to concentration. We put the embryos in culture for about 5 hours. If you remember that paper that came out, said that 6 hours was about the peak, but we want to get this all done in 1 work day. So if the technician can put these embryos in culture by between 9 and 10 in the morning, they're then electroporating the guide RNA and Cas9 complex sometime between 2 and 3 in the afternoon. They then wash those embryos and put them back into the virus particles for an overnight culture. And so this can all be done within a given 8-hour, normal 8-hour time frame. The next morning, they wash off the 2-cell embryos of the virus and then transfer those embryos, excuse me. No virus particles are transferred. They wash off the virus particles and transfer the 2-cell embryos into recipient moms. The rest is the same. Mice are born. We genotype and sequence the founders. We use the same procedure for mice and for rats. Even though rat embryos, rats have a slightly longer gestation period, we feel and have discovered that a 5-hour culture is enough for rat embryos to uptake the plasmid-based DNA. The total project cost is slightly higher than when we did microinjection. We have to include the cost for the AAV production, of course. We do use less donor females because this is so efficient that we don't need as many embryos as we would for a microinjection process. We don't need to transfer as many embryos, so we don't need as many recipient moms. We do have a higher cost in housing because we just are producing so many animals here. And we're breeding more lines. And so I'll explain that in a little bit. So in the last 8 months, so this year, Vector Builder has supplied us with AAV for 12 knock-in projects. We have an average of 92% pregnancies. Of those animals, we got on average 26 pups. From those 26 pups, we have a 34% knock-in rate. We are averaging 8 founders per project, which is actually too many. It is too efficient. So now we are scaling this back even more because we don't need 8 knock-in founders. So remember, we sequence our founders. We know that these founders have the gene of interest knocked in. Okay, so we are very, very happy with how this has worked over the course of this year and are very happy with our partnership with Vector Builder to supply the AAV particles that we need and the gene edited animals that are being born. But we all know that gene editing does have complications to it. This paper came out in February, and this paper looked at long read sequencing to look at concatenation and backbone integration following AAV-driven electroporation of CRISPR RNP complexes into mouse zygotes. So this paper goes to prove that labs really need to investigate the mice and rats that are born that they use for their research. And my core facility believes that needs to happen in-house. We can't hand off animals and expect labs to do this. And this paper, surprisingly, all of their AAV also came from Vector Builder. They used 3 different projects in varying size from 700 base pairs to 2.5 KB, varying rates of success, and that probably is in line to the size of these different projects that they did. So they had success in generating founders, had germline transmission, sequenced them, everything looked great. But then they took 5 mice. So shown here is the breakdown of the 5 mice that they did long read sequencing on. And these 5 mice, 2 of them had concatemerized. So that's a problem. That's a problem. If you have tandem repeats, that animal is going to be very, very different than an animal that has a single insertion. And so what this paper showed is 60% of the animals they looked at were accurate. And this is something that we as a core in Wisconsin feel that this is something we need to integrate into our pipeline. And we can't rely on labs to do this sort of analysis. And so what we do is we validate mouse lines now by long read sequencing. And as I told you, I have a very fortunate to have a gene editing core, bioinformatics people who are very skilled, and a DNA sequencing core in-house. So our pipeline is we take the founder samples, we do PCR-based sequencing. We breed those. If it's a B6 project, we breed it to a B6. We sequence confirm the F1s. We now keep those F1s in-house. We used to hand those off to labs, but now we keep those knock-in F1s in-house. We back cross it 1 more generation. We do PCR detection of the F2s. Once we get that far, we then take the parent. We take the F1. We collect a blood sample. We then do long read sequencing by ONT to identify any random insertions, partial integrations, anything that's a tandem repeat. And it's kind of early in the series for this, but we have validated 1 Vector Builder AAV project. We looked at that and it was correct. I told you in the beginning of my talk that we did a couple of other sources for AAV, and we've had a couple of other animals generated from other virus particles. Those also were correct. We did have 2 animals out of 4 that were incorrect. Again, our sample size is low here, but this is now our go-to pipeline here in Wisconsin. And so then we hand off F2s to the labs that need these mice. And so with that, I will take your questions. My email address is up there. Feel free to email me. My QR code goes to my website. And I hope you enjoyed my talk, and I would take questions at this time.
All right, fantastic. That was a great talk. Thank you so much, Kathy. That was wonderful. We have a couple of questions already. So one of the questions is actually about the specifications and design of the inverted microscope transfer setup. I'm guessing that's not really something you can talk through necessarily right here, but are there any kind of key tips that you can give?
So our setup is in our basement. It is on a floating table. Any vibration will cause vibrating of the embryos and the needles. And so it's very important when you get the system set up that you have it in an area that has limited vibration and somehow, like I said, a floating table or something. The setup we use is an Eppendorf microinjection setup. We have the manipulators, the joysticks are Eppendorf. We have a automatic injector, which is also Eppendorf. excellent quality, excellent products that we get from them that we use. Our needles are all pulled in-house. We make those. We have a needle puller. We pull the glass ourselves. Hope that
And
answers what they needed. Yeah.
great, excellent. And then there was a question about the specific kind of difference between microinjection and either electroporation or AAV plus electroporation. I'm sure with microinjection, it kind of started out using just microinjection of the plasmids
Yep.
encoding the CRISPR components.
Yep.
So did you see this like a difference in efficiency when you would do microinjection plasmid versus RNP?
Yes. Yes. So the needle that is used to do microinjection is microscopic, of course. And so plasmid, you can imagine, the bigger the plasmid, it can clog the needle. The purity of the plasmid, that DNA needs to flow through that needle and not clog it. And so when we're just doing RMPs, that's not going to clog a needle. So anytime we have DNA within the solution, we run the risk of just having the challenge of the purity being an issue. Again, we outsource all of the plasmid production, the guide RNAs. We outsource all of that and are very particular of where we get our reagents from so that we don't run into problems with either embryonic lysing or clogging of needles.
Mm, perfect. And then our next question was, do you use ONT whole genome sequencing? And if so, how much does that cost per mouse?
Yep, good question. So yes, that is what we use as whole genome sequencing. So this is something we developed in-house, and the price right now I think is $2,500 per mouse or per sample. And that is everything, including my lab taking the blood sample to you getting a report in the end that says what the reaction or what the reads are. Okay, so right now so it includes all of that, but that's also with us creating the animal. So that means that the design person has already been involved. They know what the design is. They know how to look at those reads for that particular project. So if you have an outside sample, we could probably do that long read sequencing, but I don't know that we could do that. It would have to invest a little time in doing the bioinformatics for you.
That makes sense. Perfect. Our next question was about concatemers. Can you screen for concatemers of AAV with PCR?
So this is something we just did. We're doing qPCR and digital PCR and trying to see if we can detect concatemers that way. Very early, we think we can, but I'm not going to say that 100% that we could do it through those techniques.
Okay, perfect. So fingers crossed.
Yeah,
Because that would be a whole lot easier.
right, exactly. That's what, or at least get some idea from the founders without having to breed all the way to F2s. Yes, we're looking at a couple of other ideas instead of waiting and doing long read sequencing.
Great, great. So great talk. Do you see any chromosomal rearrangements in your CRISPR edited embryos?
Not very often. We have to a small degree. And so not only do we do mice and rats, we also do a small amount of swine work. And so for swine, this whole AAV doesn't apply. So that's why I didn't mention them until now. And with swine, we have to do microinjection. And we also do microinjection into the cytoplasm using mRNA. And we have seen some chromosomal rearranging in some of the swine that we made. I'm hoping that these AAV techniques will minimize that as being a problem.
Okay, perfect. And then actually that leads on to the second part of this question. Have you tried mRNA to deliver CRISPR components as opposed to RNPs?
Yep, that was, 8, 10 years ago, we did mRNA. Yep, that's what we used to use. And now
Funny
we
how
do.
the
Yeah, go ahead.
oh, you just do, sorry, you just do RNPs now?
Mm-hmm. Mm-hmm.
Perfect.
Yeah.
It's kind of
Yeah.
funny how the techniques and the technology kind of follows the central dogma. We just do plasmid
Yep.
initially, then RNA,
Yep.
now RNPs.
Right. Right. And that's the whole idea is, we're being a core facility and producing a product, and we have got to be ever evolving. We can't stuff. I've been doing this for 30 years, and stuff we even did 5 years ago is kind of becoming obsolete, um, because we want to do it cheaper and faster. And we need to deliver a product to the labs, and the faster they get those mice, the more research that can be done with them.
Absolutely. Our next question is, can the electroporation method introduce a gene that is about 300 kb long?
No, no, no.
There you go.
So,
No, that's
um,
huge.
so anything over 4 or 5 kb has to be done by microinjection. It just doesn't get into the embryo. So we recently did a back that was about 200 kb, and that we did through microinjection, and we have a few animals that carry that long 200 kb piece of DNA. You can't do that through microinjection. It'll never go in.
That makes sense. Yeah.
Okay.
Many thanks for the impressive talk. Our next question, please let us know a bit more about flex viruses, which can transfect free mice, and any technical details that kind of apply to that sort of
Yeah,
set.
I guess I'm not so aware of flex viruses, so I'm not sure that I can answer that question right now.
Okay, so this particular person has an astrocytic Cree, and they're crossing with flox mice,
Mhm, mhm, yep.
GCAMP6. But we
Okay.
also want to mark astrocytes with red fluorescent protein for tracking morphology simultaneously with calcium. Um, and they plan to use an AAV, but is it better to microinject it? Yeah.
So if you can find a core that can take your flox mice, and I think I'm understanding this because you read it kind of quickly, take your flox mice and tag it. I think that's what they want to do. So yeah, that's what I mean by taking a customer's mice and taking embryos from them that are already carrying an edit, and then introducing a second mutation on top of that. So yes, you can find a core to do that. It is possible to do that. Mhm.
Okay, great. And then more of a kind of a logistical initial design question. If you're wanting to get really just initially into in vivo mammalian model studies, if you're choosing between, let's say, mouse and rat and swine, are there any kind of key considerations that people should start with when choosing one of those three models?
Oh, sure. Good question. Um, it really depends on the lab setup. Obviously, swine require staff and a farm and an area to house them, and are much more costly to maintain. Mice and rats are pretty similar in term. We have our mouse room right next to our rat room. We don't have them in the same room. We have staff. Everybody can go back and forth between the animal rooms. The care staff is the same. Some people like rats for certain physiological studies. A lot of cancer researchers like to use rats. There's a large need in cardiovascular research, hypertension. Um, those types of experiments are better done on rats. We worked with a transplant surgeon here at the UW who did liver transplants, and they liked rats to do their transplant surgeries on. And so for varying reasons, people choose different animals for their research. And so it's really a case by case basis as to why you would choose one species over another.
Very interesting. And then there is another question. Wonderful talk. Have you seen a difference in concatemerization rates between AAV1 and AAV6, or have you only used
Yeah,
AAV6 for your
we've only
purposes?
used AAV6. That's all we've used. So, and I think from what I'm hearing from other cores, everybody kind of uses AAV6, from what I understand.
Okay, perfect. And our next question, for a cancer study, which do you think is more efficient in the physiological expression of an oncogene? Is it ectopic implantation of CRISPR engineered cells
Mm,
or making these CRISPR engineered transgenic mice?
yeah, yeah. That's a case by case basis. So when you inject cells that have been engineered into an animal model, obviously that is not a systemic approach. You're not going to get that from generation to generation. You're going to get that on a per animal basis. And so um, if that answers your biological question, that's great. If you want um, a cohort of animals that are all genetically the same, that you can then apply a drug or something to, then you need an animal model that is genetically able to transmit that from generation to generation. So it really is on the scientific question that you're trying to answer as to which approach is better.
Makes a lot of sense. It's kind of the scope and scale of
Yeah,
the size of the study almost and
right,
reproducibility. Yeah,
right, exactly. Yeah.
that makes a lot of sense. Perfect. Um, and then I believe our final question is uh, based off of the kind of micro injection low efficiency versus something like AAV. Um, do you think I'm assuming these are not considering viability of the embryo. Do you have any guesses about why the efficiency is so low for microinjection compared to other approaches?
Yeah, I think it just does too much damage to the developing embryo because it's physically causing, interruptions within the cytoplasm and the nuclear membrane. Um, as you can tell with electroporation, the exposure to the voltage is very, very brief.
Hmm.
And I think just time frames, the embryos are out of the incubator longer. They have to sit on a microscope. Why we do all of that manipulation. We usually put out batches of 50 or so embryos, and then we manipulate them, and then it takes time to do that. And so they're out of the natural environment of the 37 degrees, 5% CO2. While with electroporation, you take the dish out, you rinse, you load it on the slide, you zap them, and you get them right back. So the turnaround time back into the incubator is very short. And so I think for various reasons, the viability is just something that we just have to accept with microinjection.
Hmm. That makes a lot of sense. Perfect. And then we had one last question sneak in. For virus purification, is there a particular purification that you prefer when you, for instance, order viruses from Vector Builder?
Yeah, so from my understanding, we just order what are called pilot study um, AAV, and those work great. So
Perfect. Excellent. All right, great. Well, we're finishing right on time. Thank you so much again, Kathy. It was a wonderful talk and a fantastic discussion.
Yes, yes,
And
we had great questions. Thank you, everybody, for joining us today. This was really nice to be able to do this.
Perfect. Thank you all so much. And we hope that you can join us for the next edition of our Gene Delivery Seminar Series, which is happening on November 19th. And we will be sending out announcements for that in the coming weeks. But thank you so much again, Kathy, and thank you everyone for joining. And this video will be available on demand as well as on our webinar page, which will be appearing on our website shortly. So thank you again.